All right, everyone. As you get settled in back from your short coffee break, I do want to mention that the Congress Center is offering lunches for purchase in Hall 4.1, the first floor, where the posters are located. And so lunch will be available for purchase between 1 and 2 p.m., especially if you have an afternoon tutorial that you're attending. So now I'm going to talk a little bit about pseudotime and trajectory inference. So pseudotime, what does this mean? So more generally, these are referred to as trajectory inference methods, and this can be over what we think of as time or space or any kind of dynamic dimension. The idea is that cells can be computationally reordered based on the similarities of their transcriptome profile, so based on their gene expression. So what does this mean? So suppose that we expose cells to some signal, like a differentiation factor, so we tell the cells to start differentiating. And so this is going to start some dynamic process of the cells. And we expect the cells are not going to respond exactly identically, right? Some cells are going to respond a little bit faster, and some are going to respond a little bit slower. But if we collect cells all at the same exact time, and we do a computational reordering, we would expect that the faster cells, they would have gone through the progression that we see the slower cells in. And so this is the idea of ordering things in pseudotime. So we are able to see the slower and faster cells, which gives us an idea of the whole dynamic trajectory that the cells are going through in response to that stimuli. So here's kind of a general idea. So suppose here on the left, here's just a population of cells. And let's say I have gene A, and I'm representing the expression of this gene using this different colors. Maybe this is a high expression blue, and red is a low expression, or vice versa. And so you can see the expression of this gene, has some distribution. And so if I wanted to reorder the cells just based on gene A, maybe if I just put the cells in some order, this doesn't really look like, it doesn't really order according to the expression of gene A. And so you can kind of, shuffle things and you get a little bit better. And the idea of a computational reordering is we'd want to get something like this. We'd want to see that the expression determines the order of those cells. But in reality, we don't have just one gene. We have 10,000 or more genes. And so we're trying to reorder over this high-dimensional data set. And we also don't necessarily always have a linear trajectory. So it's possible that the cells are going along some trajectory, and then they split into two separate cell types. And so this would be a branching point or a tree structure that we would want to observe in our data. And then sometimes we might see something that looks more like a cyclical pattern. So you could think about cell cycle, right? It's not just a linear trajectory or branching point, but something that the cells are going to revisit over time. So there's actually more than 50 methods for pseudotime reconstruction. If you go to this GitHub page, it lists all the different methods and has links to them. And recently there was a massive effort to do a benchmarking of all these methods, and that's published just maybe a couple months ago. And so that has recommendations for how do you choose these methods. So I'm only putting here just one figure from the paper, but if you want to know kind of a guide to choose how to reorder your cells, I highly recommend reading this paper and going through the different figures they have. So the one guide they give is, do you expect to have multiple trajectories in your data set, or do you have one trajectory? And then from there, do you expect a particular topology? So do you expect that linear reordering, or do you expect there to be a tree structure or a cyclical structure? And then based on that, they'll give you these different options. And then here's some of the different methods that will do this type of reordering. So if you just want a linear structure, these 4 methods are able to do that. And then they also rank the methods by their user friendliness and their performance. And so the performance usually has to do with speed, and then the friendliness is how easy is the package to use and to get your data into. And so this gives you a lot of information about choosing that method that you'd want to use for your particular data set. And so I'm not going to go through all 50 methods. I'm just going to choose some of the ones on this list that they said generally perform pretty well across a variety of data sets and scenarios. Okay, so one of the main differences in the methods is their ability to infer specific topologies. So that linear topology, one of the methods is called TSCAN. For more of a tree-based or branching trajectory, we can use a Slingshot. And then for a partitional tree where we have maybe disconnected trajectories, Monocle 3 or the PAGA, our package, are able to do that. And so again, on that GitHub page, we have links to these methods as well as that paper, the benchmarking paper. And so all these methods, these ones and the previous ones, they all implement some preprocessing to remove noisy or lowly expressed genes or lowly variable genes. So you're just going to use those genes that have a more informative expression, so highly variable genes in your data set. So additional differences between methods are that dimensionality reduction is done via PCA, TSCAN, or UMAP, or diffusion maps. Different methods are going to use different implementations. The type of graph or tree is inferred on the cells themselves. So some of the first methods try to draw a tree connect every single cell. Some of the later ones, they try to just cluster the cells and then draw a tree between the cluster centers to kind of help eliminate the variability due to noise. And then the amount of supervision that is needed or able to accommodate. So some methods will need like a starting point, or some will let you put a starting point if you prefer, for example. And so the first method I want to talk about is TSCAN. And so this is a linear trajectory method. And so it first actually reduces the dimensionality using PCA, and then it will cluster the cells. And so it actually uses mclust to cluster, but you could implement, it kind of lets you use whatever clustering method you want to use and just feed in the actual cluster identities. And so here I'm visualizing the PCA dimensions of these cells. And this is the data set I'm going to use for all my examples today. So this is a data set where just some embryonic stem cells were exposed to differentiation factors and then collected at different time points. So they were collected at hour 12, hour 24, 36, 72, and 96. So I have these 6 different time points of my single cell data. And then the clustering algorithm that was used here separated some of these, like 72 and 96, into their own separate cluster. But we're still able to use this information. So you can see that the clusters that the TSCAN implemented here match pretty well with the original identities that we know about. And then from there, it fits a minimum spanning tree to connect the cell cluster centers. So this is different than the idea of connecting every cell. Again, that would often produce really noisy pseudotime estimates. So the idea now is if you do the clustering and then do the minimum spanning tree along the centers, then it's less prone to deviations just due to noise. And so once you connect these different centers, you can see that here I didn't even require that it started at cluster 1 and ended at cluster 5. But if we go back, we know that cluster 1 is my first sample, which was hour 12, and then the 6 was the 96 hours. And so it got this order pretty well calculated. And then the pseudotime, how we obtain this. So the way that we actually get each cell to have a time is it projects the cell onto this tree, and then we'll to the nearest branch, and then it will give a pseudotime for each cell. So it's just projecting each cell into the nearest branch point. And so once we do that, we then get this pseudotime ordered for every cell. And then for here, I'm projecting just plotting a particular gene, so this CCDC80 gene, plotting the log expression of this gene versus the ordered pseudotime from the TSCAN method. And you can see the dynamics of this gene changing over time. And so just to be consistent, each of the methods that I show, I'm going to plot the same gene, just so we can see how they're performing differently. Okay, so building on TSCAN, which is pretty simple, is Slingshot. So it's very similar. It's going to use that minimum spanning tree just like TSCAN, but it's going to calculate different distance metrics, and you can actually implement your own. So it doesn't have to be Euclidean. It can be the Manhattan distance. It's a little bit more flexible and lets you pick those options. It also lets you choose a starting point and ending point if you choose. So here I can choose to start at the cluster that has 12, and the ending point needs to be the cells that have the identity 96. And it'll fit that minimum spanning tree the same. So this is the kind of the straight line here. And then to get pseudotime, it'll fit an arc, kind of like a principal curve here through those same points. And it'll project the cells onto this principal curve now. And so the idea of that is that instead of just having to fit onto these straight edges, it gives a little bit more distance between the cells for the pseudotime estimation. And so if we look at one of the figures from the Slingshot paper, so in the example I'm using, I just have a linear trajectory, but this is more amenable to having tree-based topologies where you have these splitting points. So here we have this dimension reduction, so this could be PCA or TSCAN, and we see that it looks like there is some kind of branching happening if we think the starting point is this orange cluster, and so it'll fit this minimum spanning tree here, and then it'll fit the principal curve, and it can fit through both trajectories. And so, for example, these orange cells, they actually belong to both trajectories, and you'll get two pseudotimes. So you'll get a pseudotime for this branch here, and you'll get a pseudotime of this branch over here. And so that was represented in 2D. And then in 3D, here's an example where, again, we have these cells represented on this PC plot. And you can have multiple trajectories, and each cell can belong to more than one trajectory. All right, so again, for Slingshot, it's fitting that principal curve through each lineage, and then it will do an orthogonal projection for each cell onto that curve to get the pseudotime for each cell. And so again, if I plot the same gene as a function of pseudotime, you can see it's increasing over time, very similar to TSCAN, but some of the options of using this method are more flexible. Okay, so the other thing that Slingshot offers is that it was based not just using PCA or T-sign or UMAP, but diffusion maps. So this is another type of nonlinear dimension reduction based on those local similarities. And so you can use that diffusion map reduction, and it works pretty well as the PCA and TSNE. And so you can see from here, it looks very similar. This particular gene, once you get the pseudotime reordering for here, I mean, it looks very similar to the PCA-based pseudotime ordering. But again, it allows you to have all these different options where it's more plug and play of what you want to use for that particular data set. So now moving on to Monocle 3, so kind of making things, getting a little bit more structured here, more complicated. So Monocle, the first version was building, connecting every single cell, and that didn't typically work too well, so then they moved to Monocle 2, which I'll talk a little bit here, and then Monocle 3 is the same as Monocle 2, but they borrow some ideas from Paga, and they allow for disconnected trajectories. But the Monocle 2 and Monocle 3, the base is very similar. So they actually reduce the dimension using UMAP, and then they cluster the cells using a type of clustering that kind of will push each cell closer to each other. So I'll show you in the next slide. But again, I'm representing that same data set I'm using every time. And then their clustering method, again, breaks some of these apart. So my hour 12 actually is now these two different clusters represented here. I gave them the same color since we know that they're the same time point. So the way that their clustering method works here is that you have your reduced dimension. So this is using their UMAP, or you could use PCA, for example. And then it'll try to guess an initial cell trajectory on centroids. So it'll say, well, let's put a centroid here, and then some other ones here. And then it'll try to push in the points toward the centroid here. So you can see it's pushing in these points toward the centroids. And then based on that, it'll update where they think that centroid is. So it updates a new location based on the distances of these cells. And then it'll project back to the high dimensional space, the original space, and then reduce the dimension again iteratively until it reaches some convergence. And so then the idea is, again, since this allows for more tree-based topologies, you can select like a root node and say this is the starting point, for example, and then say that you want the distance defined from this root to the end, and also it'll define pseudotime from this root point to this branch. And so notice here that that if you just select the root on this in particular, it doesn't give these two different pseudotimes. So even though this has two branches, it gives pseudotime as a function of the whole topology. But you can say that this is a branching point. So the pseudotime is defined over this whole structure, but then you can say, well, there's a branching point here, and you can kind of look at each of them separately. And so here's an example on the same data set. So when I was trying to make this, I wasn't able to get 12 to connect in order through 24 and 36 in order. So I said, well, let me try the disconnected tree and see how that works. So then here I just did the disconnected tree on each hour individually. So what I'm doing here is I'm actually getting pseudotime for this cluster of cells from hour 12, and I can get a pseudotime projected onto this tree here, and then pseudotime independently for hour 24, for example. And so if you look at the actual pseudotime that it assigns, you can see that it's based on within each individual piece here that I made. So if you use those projected pseudotimes, what you end up getting is something like this. So it looks a little bit messy. But what I realized you can do is since I know the actual order of these, you can rescale the pseudotime so that I know this one comes before this one, for example. And so when I do this constrained pseudotime within the true times, we do get something that looks very similar to those other methods. So again, I think Monocle 3 is really useful if you have those disconnected trajectories and you want to be able to do that. But again, these methods are kind of building on each other, becoming more complicated. And so if you do have some more simplistic structure, it might be more useful to use something that was made for something more simplistic. Okay, so one of the things that we do after we have our pseudotime, and so again, those are the most commonly used methods that I just went over. But once we have those, we want to look at, well, how is a gene expressed across pseudotime? So we can get our, for example, our Slingshot pseudotime and plot the expression of a gene and see how that's changing over time. So maybe, we think that this gene, starts out, at 0 or very lowly expressed, and then over time, or pseudotime becomes more highly expressed. And most methods are fitting like a generalized additive model, a GAM with like a low S, and then you get this nice figure to look at. But what do you actually do with this information, right? So you can visually compare the trajectories, but it doesn't give you a lot of quantitative information that you can use later on. So one thing that I was suggesting is that you can use what's called trendy. So this was a method that I developed for any type of ordered high throughput experiment. But if you think now that you've ordered your single cells, then you can place that into this format. So this was designed for general type of time course experiments, but if you have an order to your cells now, you can also apply this. So it'll fit a segmented or piecewise regression to each gene, and it'll choose the optimal number of breakpoints and the location of those breakpoints. So imagine here you have time, and so this could be real time or pseudotime, and you plot the expression of your gene, and there's these dynamic changes that happen at specific time points, and so it'll model what time those occur, and then you can also look at the rate of change and the direction of change. And these are quantitative outputs that you can then compare across different genes or across different data sets easily, for example. So here for trendy, we're summarizing time. So again, I put time in quotation because it's pseudotime, and the magnitude of gene expression changes. So this is again this row B3 gene that I just showed with the GAM with the low S. And so here I can say, well, this gene looks like it's generally increasing at a given rate until pseudotime 9, and then after that it's increasing at another rate, right? And so then you could get this information for every gene and say, okay, well, a lot of genes are changing around this pseudotime. Does that correspond to something specific in my experiment, since I know that some of these cells have labels? And then I can say, well, are there genes that are going up first or down first, for example? And so here's an example of all the gene trends. So here's each individual gene, and then if it's going up at first, it'll have this red trend, and then if it starts going down, it'll have a blue trend. And so you can see most genes tend to be going up first in this just very simple example. There's a number of genes that are going down throughout the whole pseudotime, and there's some genes that kind of start off the same, and then they start changing after a given time point. So we can look at some of those genes individually. So here I'm looking at some dynamic or temporally expressed genes using trendy. Again, these are genes that go up first, so you can see they're all kind of going up first over time and then have different patterns along the time course. So this T gene, which is highly involved in development, so this is one of my scientists' favorite genes. And so this one tends to go up first very quickly, and then it goes down and kind of turns off after a while. And then we can look at genes that kind of stay the same first. So some of these kind of start out at just 0, and then they start to go up, and then maybe they're kind of going down or more stable over time. So these all correspond to maybe different biological patterns that you can compare. And then also I'm showing genes now that go down at first, and then some of these kind of go down and then go completely silent, for example. And so these are all different biological patterns that you can look at using pseudotime and using trendy. And actually, on the GitHub page, I linked to a tutorial where I just went through this example using the same data set, so you can kind of get an idea of how to do this in practice. So for pseudotime, I think I was going to end here, and I wanted to save some time for questions before I move into the immunology section. So if you guys have questions, please, you can come to the microphones. Or I'll switch slides if there's no questions. Okay, running good on time. Okay, so I'm going to transition now into talking about single cell for immunology or immune profiling. So why immune cells? So the immune system is comprised of a diverse set of adaptable cell types that are responsible for responding to changes or maintaining a healthy system in the body. And so single cell technologies offer us the ability to study the cellular heterogeneity in an unbiased way. And so this means we can discover new cell types or cell states or cells transitioning between cell types, for example. And so I wanted to briefly mention cell type classification. So we talk a lot about getting these clusters and visualizing them, but we don't necessarily know what type of cells these are. And so if we want to study cellular heterogeneity, we need to say, okay, well, this is a group of T cells, but then this group over here, it maybe looks like a combination of some type of cells, and we want to say it's a new type. We need to know what else is already there. And so the way that this is usually done is super time-consuming and difficult. So usually we'll find genes maybe for this green cluster here that are different in this cluster than all the other clusters, right? So we're computationally finding marker genes. Or we might find these cluster-specific differentially expressed genes. But then we have to go and take that list of genes that we got for that cluster and compare to our known list of marker genes that we have from prior experiments and knowledge. But even if you have that domain knowledge, it's still very time-consuming, and it's hard to really put all that information together at once to determine that this is a specific cell type, and we're sure that that's the cell type it is. So there's some recent work into doing automated classification. So this can help the process and make it a lot faster. There is a benchmarking paper on bioRxiv. It's still relatively new. It looks like it's being added to. But it does actually list all the methods that have been published thus far and some of the ones that are still in bioRxiv. So it does have a reference list to look at. But it was trying to evaluate the different methods. And all the methods do require like a labeled training classifier or a labeled training data set or a pre-trained classifier or a list of marker genes that you give it. And so I'm not going to go over all the methods, but this one, SingleCellNet, they're all using some type of learning or decision trees. But the reason I didn't go over all of them is this paper kind of says that they all perform pretty similarly. So SingleCellNet, this requires a labeled training data, and it uses a random forest classifier on genes that they determine to be the most suitable for classification. So it determines genes that they think are going to be the most useful in classifying. And then what's useful here is it outputs a classification score of new cells to existing cell labels. So it doesn't just say that these are T cells, but it says this is the score that we think, for example, this is the score that we think our cluster F is a B cell cluster. And so then using this, you might be able to say, well, I think, let's say you had one of these that was half and half, you might say, well, this is a transitioning cell type, for example, right? If it was, naive T cells and maybe activated T cells or memory T cells, you think that it was half and half, and that might be a transitioning cell state that you were able to capture. But again, you have these different clusters, and you're going to compare them to known clusters. And someone had asked, how do we have that information before? And the answer is that previous experiments we've done like sorted cell populations. So like if we know marker genes for our NK cells, then people have done flow and done like sorted experiments where we know those are all NK cells, and then that's how we get those purified populations that are labeled. And there have been people that have, like for the tabular muris, for example, that all those mouse cells that they actually went and spent a lot of time curating what cell types those were. So if someone has gone in and spent a lot of time doing this, then we do have some access to that information. And so this paper actually is still in bioRxiv. So I was going to mention that a lot of the classification or automated identification methods are still in bioRxiv just because this is such a new thing. It still has a lot of room for development because we had to first get all the cells and that prior information to be able to do this. So this is an ongoing area of development, but I think it'll be really useful once these start to be useful. People will want to use them because everyone gets this and then wants to know what are the cells, and then you spend so much time just getting those labels on there. Okay, so the next thing for immunology is CiteSeq. So this was also mentioned briefly. It's a multimodal method that you can obtain not just the transcriptome, so the mRNA, but also the cell surface protein markers. And so this is an experimental technique where you have a poly A tail and then a tag with an antibody that attaches to a specific protein. So the experimentalist actually decides which proteins they want to go after. So if you want to go after CD45, then you get an antibody for that specific protein. And because this will have a poly A tail, once it gets into these different droplets, it'll be sequenced just like any of the other mRNAs. And so then for each cell, you'll end up getting the expression for each gene, but you'll also get the expression of that protein marker that you went after. And so this experiment, you can go after just a few protein markers that you're interested in, or maybe up to, I don't know, I haven't seen more than like 40 or 50 actually been used. And a lot of that's because the experiment becomes more expensive as you add these different antibodies. So the benefit of this, though, is that it allows association of the protein levels with mRNA for the same cells. And the protein has less of a dropout problem. So if there's, if you have marker genes that you know for specific cell types, but it's not a super highly expressed gene, and so it'll have some dropout in the mRNA, then for protein it might not, and it might help you to be able to classify those cells a little bit better. And so again, it can help with those subtle differences in classifying cells. Here's actual an example from the original paper for CiteSeq. So here they looked at a cluster of what they thought were NK natural killer cells. And so if you looked at the mRNA expression of these two markers, of these genes, CD56 and CD16, you can't really tell much difference happening in this cluster, right? It just looks like this is highly expressed and this is lowly expressed. But then the protein, so what they call ADT, which is the antibody-derived tags. So here they have the cluster, and it looks like there's a subset here that's more highly expressed in the protein, and then CD16, this area is also more lowly expressed. And this actual dual expression here is actually a known subset of NK cells in the literature that they were able to find just based on those protein levels and not in the mRNA levels. And so that's kind of just showing how having both of them can benefit in your cell clustering and classification. Okay, so another reason immune cells are really interesting is so that they have what are called, there's B cells and T cells, and those have what are called receptor chains on their surface, referred to as TCR and BCR. So that's, here's a T cell and here's its receptor chain. And these are just protein chains. And so the protein chains that are expressed on the surface of a T cell are actually mostly identical for the same T cell, but across different T cells are extremely diverse. And the reason for this is that there's a recombination that happens called VDJ recombination. So here's a figure from 10X where so each of these little chains here, there's actually two of them. So for T cells, there's an alpha chain and a beta chain. And then there'll be this VDJ recombination where somatic recombination where different rearrangements of these genes will be made, and then they'll express a specific protein sequence that gets expressed on the surface here. And so what's interesting about this is that each T or B cell and its progeny will have a unique nucleotide sequence. That's called the clonotype. And so for T cells, they get activated once they encounter like an antigen or some type of foreign molecule. And so when that happens, they'll actually expand and proliferate. So they're going to make more copies of themselves. And for T cells, they'll actually have the same clonotype. So if you were to sample single cell and you see T cells that have the same clonotype, then you'll know that it was responding to something specific. And so expanded cells will share the same TCR clonotype. For B cells, it's a little bit more complicated because they have a lot of mutation that happens, so it doesn't work exactly like this. But then also for T cells that we know when they're being developed in the bone marrow, that the TCR beta chain gets recombined first, and then there's an expansion that happens there, and then the alpha chain gets recombined, so we'll actually know which ones are related early on if they have the same TCR beta but a different TCR alpha chain. And so we can use these relationships to kind of study responses that are happening in the body, only since we're able to do this at the single cell level. Okay, so how do we actually get this information? So if we use the SingleCellNet methods, we are actually sequencing those genes, right? So that protein sequence that's expressed on the cell surface is representative of that gene sequence in the mRNA that's being expressed. And so if we're using a full-length single-cell method, we can actually get this information without even adjusting the standard protocol. The challenge is that because this is so variable and it's changing all the time, we don't have a good reference, and so it's difficult to align and distinguish sequencing errors from real rearrangements. So the first method that was able to do this is called tracer. So what they did, they did their single cells and they get their reads for each cell, and then they'll align what's called the TCR reads to a combinatorial reference. So they took all those VDJ regions and shuffled them around, and then try to align reads to those shuffled rearrangements. And so that had to be a lot of rearrangements that they made for all these different combinations. They align the reads, and then once they have them aligned, they'll construct a contig to get the full TCR sequence. And once you have that, then you can actually compare across cells. So once you have the full TCR sequence for a particular cell, then you can compare, for example, just the V part between different cells. And then once you have the full contig, then you'll want to realign your reads. So add that contig back to the original reference, and then realign your reads so that you can actually determine the quantification of how much that's expressed at. And so this is the realignment step. There's also a step where you have to determine whether the contig that you get from the short reads is actually translatable. So this is called determining the productivity. So basically, if you get your contig, you want to make sure, for example, there's not a stop codon right in the middle. So it's kind of just checking that what you got from the short reads and reassembled is a reasonable product. Okay. Second method to do this is called VDJPuzzle. And so it's kind of pretty similar. So again, they're going to align reads against the reference genomes, but they're not creating a combinatorial. They're actually just aligning directly to to each of these separately. And they'll do a de novo assembly of the whole TCR contig. And then they'll put this through IG blast, which kind of checks again so this is an immune specific blast, and it'll check that this is a reasonable contig, and they'll align back to this in case that some of the reads were kind of on the junctions, and they'll get a new TCR assembly and then rerun this again to make sure that it's a productive contig, and then they'll get their final set. And again, once you have the final set, for each cell, you can compare the regions across different cells. And so you can see, where the shared TCR clonotypes, or, do any of the cells have the exact same ones, for example. Okay, so that was with the full length, that method. But, now that people are using 10x and wanting to get this high throughput, how do we do that? So they actually adapted their protocol so they're able to, in the same experiment, obtain tag-based RNA-seq and the full-length VDJ sequence. So they use a targeted PCR on all the BCR and TCR gene regions, and that's able to retain the full-length sequences just for those specific genes, and also retain the cell and UMI barcodes for that. And so they're doing something a little bit different. It's actually similar to VDJPuzzle that they're using to do the de novo assembly to handle the diversity when they're realigning. But they do have the advantage here that they have the UMI barcodes and can make sure that they have a consensus for that cell. Okay, so types of analyses that you can do with this. So the first is a diversity quantification. So you can look at, for example, the clonotype length, or the composition and quantity, or the expression level. So here's a figure here where this is actually a tumor, and this is another tumor. And so the clonotype, again, that's the unique nucleotide sequence for each cell. And so and this is the top 100 in this tumor, and so even the one that's the most common is still only 0.02%. So again, we have this huge diversity just because of all these rearrangements that happen, but even if there's an expansion and some of them have the same, it's still a very small percentage overall. And so you can also look at, what are the V, D, and J alleles separately, and it's called the immune repertoire when you're looking at all the clonotypes together. You can look at the most frequent combinations. You can also look at the total diversity within an individual, or looking across individuals or across tumors, for example. You can see if any of these clonotypes are shared across tumors. Another type of analysis is looking at how the cell type clustering corresponds to the clonotype clustering. So here on the left is clustering of T cells, and each color here is a subtype of T cell. And then on the right here are maps of shared clonotypes. So again, because there's so much diversity, it's not as much to see. So for example, clonotype 1 in red here, each of these is over here, but you can see the main population is kind of highlighted right here. And if you look on this map over here, it's kind of shared between a number of different T cell clusters. So again, when T cells share clonotypes, it means that there's some activation that happened. It was recognizing a foreign substance, and then the T cells become activated, and they become other types of T cells. And so that you can actually see that happening when the T cells are not just in their same cluster across clonotypes. And then the next figure, I'll show more of that. So this is the idea of lineage inference, so characterizing the immune response by identifying and quantifying the clonotypes present. So the expansion represents a response to a specific antigen, and so we can identify the shared clonotypes across types of T cells. So here's a good example here on the right of this. So this is a TCR analysis during immune response to malaria. So if you have the cells uninfected, there's no shared clonotypes, and then over time at day 7, now the cells are recognizing the antigens, and now they're expanding, and so you have these shared clonotypes that are happening at day 7. So this is obviously some type of time that's happening as the T cells are recognizing that threat. And on the left here is a similar plot where, again, T cells start out as activated and proliferating, and then as they respond, they become central memory. And so if you think of this as going in this direction, you have these clonally related T cells marked in red here, and you can see that they become more common as the T cells are dividing and responding to that antigen. So this gets back to the idea of trajectory inference by shared clonotypes. So here in this paper, they actually have these 4 different types of T cells. And if you think about it starting in this direction over here and then going in this direction, this is how they ordered the cells based on the gene expression, just like I talked about in the last section. So this has nothing actually to do with TCRs yet. So this is just based on gene expression. But on the right here, now they're visualizing the cells in this reduced dimension, and they've colored based on shared clonotypes. So again, it looks like a lot of colors here because again, having a shared clonotype is still a pretty rare event just because there's so much diversity. But you can see that if we didn't think that there was this pseudo-time ordering of these different types of T cells, that they would share different clonotypes. But actually these, going from red to this light blue, has this thickness of this represents the proportion of shared clonotypes. So this has a high proportion of shared clonotypes from this cluster to this cluster, whereas from this cluster to this cluster, there's no direct clonotypes present shared between these. And so you can see that this goes to here, and then here goes to here, and then this one goes to here. So you can see the shared clonotypes by this thickness represents the T cells change, the type of T cells changing in response to that, to whatever they're responding to. Okay, so that's what I was going to talk about for immunology. If you guys have questions, I'm happy to answer those while we transition to the next speaker. Thank you. Yes?
So are you able to comment on the relative strength of Site-seq versus Site-off?
Yeah, so I'll repeat the question. So we asked what was the difference between Site-seq and Site-off?
Or the strengths?
The strength. So Site-seq, you're gonna get the transcriptome. Site-off, you're not, right? But Site-off, you're gonna get many more proteins. So it depends on what you're interested in, what you're looking at. Yeah, they have their own strengths. Yes.
For the pseudotime, I have a question. You showed the gene expression over the pseudotime, and
yes.
I mentioned you could use a GAM, for example.
Yes.
You could use the same GAM to test it against a null model if you have changes over the pseudotime, right? We would just need a clever way to pick how many breaks you have.
Yeah, you could figure out a way to interpret the GAM. Yeah, that would be a good idea to have a null model and determine where the breaks are. Okay, thank you guys.